Fantastic. All right. Thank you very much for the introduction, and thank you for all the organizers for graciously giving me the opportunity to share my work and to utilize this time in general to add more facets to the topic of diversity in science. As you can see by the title of my talk, I have a very basic biology project that I would like to share with you. But to start, I would like to tell you a little bit about myself and how I convinced myself to give a lecture in a series focused on diversity in the sciences. So I'm going to start by telling you the traditional story that scientists tell each other when explaining how they got to where they are. But I'm then going to tell you another deeper and more personal story about how I got to where I am and how certain aspects of this academic system prevents the inclusion of more diverse perspectives. So I received my bachelor's degree in molecular and cell biology from UC Berkeley. In my final 2 years as an undergrad, I volunteered part-time in the Cooper Lab in the Lawrence Berkeley National Laboratories. And I learned a lot about what it means to work in a lab, and I was really interested in research, but I wasn't really ready to commit to a PhD. I actually didn't even know what it meant to do a PhD. So I took a full-time position in the Autobi lab in the UCSF Cardiovascular Research Institute. This is to solidify my research background. I'm going to explain more thoroughly on my next slide, but even after 4 years of working in the lab, I still didn't feel ready to commit to a PhD. But I decided to give basic science another shot, and I started work at Sanford Burnham Prebys in the D'Angelo lab, which studies nuclear pore complexes in the context of cellular and organismal physiology. And so I really enjoyed my time in the lab, and I got the chance to contribute to publications and even write a first author review. Those experiences gave me a lot of confidence about my scientific ability, and I realized that I wanted to have my own project and to publish my own primary research. And if I was going to do all that, I felt compelled that I should try to go and attain my PhD. So last year, I officially started my PhD in the D'Angelo lab, and I'm excited to tell you about the research I've been doing. But before I do, I will tell you about the part of the story that comes before all this and how that led to me having a different perspective about this career than most of my peers. So I'm from California. When you tell people outside of California they're from California, they probably imagined Hollywood and LA and palm tree lined beaches. That definitely wasn't where I grew up. I grew up in a small town in Northern California called Shingletown. And when people they say they're from a small town, they usually mean one of the non-major cities in an area. What I mean is a proper rural mountain town with less than 2,000 people. This picture shows what most of the town looks like. As you can see, it's mostly trees. And I love that aspect and many unique and beautiful experiences from my childhood. But there were some big disadvantages to growing up in an area like this in relation to pursuing a career like this. I had many amazing teachers who encouraged me to reach for academic excellence, but there was and still is a pervasive anti-intellectualism pervades the culture. This concept of anti-intellectualism has become a hot topic in the last 4 years or so, but I've known it well my whole life. Additionally, people who traditionally think of California as a liberal place, but in my school we had many religious fundamentalists. And this was relatively benign in English class where Bible was taught as an exemplary piece of literature, but it was more problematic in biology when the teacher refused to teach or test on evolution. You can imagine that this made appreciating the beautiful cohesiveness of the biological sciences very difficult. And subsequently, my love for biology would lay dormant until after I left. So as I told you before, I went to Berkeley and I also lived in San Francisco, and most people would think that moving to the big city of San Francisco would have been the hardest transition. But for me, the move between Shingletown, these green trees, and Berkeley, which I found that this is a big city, was exponentially greater. It is a little hard to describe to people who grew up in cities, but the way of life is very different. It was so different than when I moved to Berkeley. I had trouble sleeping, and the constant bustle of the city and people moving all around me made me feel like an outsider, like I wasn't supposed to be there. Despite that, the struggle that has haunted me most throughout my professional career was with financial insecurity. As you can imagine, a small town like Shingletown was not filled with wealthy people. My family was not the poorest by any means, but my mother was a single mom trying to raise 2 boys, And I remember many moments in my life when it became abruptly clear that we didn't have quite enough to be able to pay for basic life necessities. So as you can imagine, no one in my family had gone to college, but I was told that if I went to college, I could achieve a level of financial security that I never experienced as a child and that appealed to me very greatly. Even though the cost of attending college did put serious strain on our finances, I did have to take out loans. I kept thinking about that job that could give me a little peace of mind, and I decided to do it and go to college. Despite the financial stresses while I was in college, I was able to rediscover my love for biology and start volunteering in a lab. You can see me here proudly wearing my first lab coat. So even after I volunteered in the lab and managed to graduate from undergrad, this is me graduating, that was more than 10 years ago, and I only recently started my PhD. And you might wonder, why is that? Well, a lot of the delay was me considering other career paths that could lead to a higher salary sooner, which might sound greedy to some people, but I think you can see from the story I'm telling you that it's a little more complicated than that. The other part of the delay to starting my PhD was from the extremely rational fear that the graduate student stipend and the subsequent postdoctoral salary would not be enough to support my mom if she lost her job. I say it was a rational fear because that is exactly what happened. My mom became too sick to work and needed financial assistance. That was a bummer, but after this part of my life, I was excited to move on to happier things. As you can see here, I got married to an amazing person. And the strength of our combined capabilities allowed me to believe in myself that I could financially survive the training phase of a PhD level career. And with the support of my loving wife, I did finally start my PhD. So I enjoyed the first year of my PhD very much, and I was really excited to see this lecture series come up. When I was first considering presenting at this lecture series, I was daunted because I've seen many fantastic presenters cover many very important issues such as racism, sexism, and I wondered if there was anything of value that I could contribute. But since this most recent movement to diversify the sciences, I've been exploring diversity, equity, and inclusion. And what I have realized is that the complexity of these issues is more broad than I had previously realized. My racial background is Caucasian and Asian, which are generally not considered underrepresented in the sciences. In the same way that people think of, but in the same way that people think of minority racial groups, feel like outsiders, I've also felt like I don't belong. And belongingness is at the center of this Venn diagram for a reason. The absence of the feeling of belonging is one of the main reasons that achieving diversity in science is such a struggle. That diversity is critical for the originality of perspectives that has the power to change how problems in science are solved, and therefore to make science a better discipline. Now, I know we won't solve them all today, but I am thankful for this lecture series to highlight the issues so that we can keep working to solve them. And I hope that serves as a good philosophical and intellectual introduction to me. And I hope that you are able to simmer on those thoughts for a little while I tell you about some of the science questions that I've set to solve in my PhD work. So to begin, I would like to show a eukaryotic cell, and I would like to highlight the structures that span the nuclear envelope to connect the nucleus to the cytoplasm, highlighted in red here. They are called nuclear pore complexes, and throughout this talk, I will refer to them as NPCs. Taking a closer look at the NPC, we see that it is a large, complex, multi-protein structure that is the pathway for both nuclear import as well as nuclear export. I will explain the structure in more detail shortly, but what is important to remember throughout this presentation is that because NPCs are the sole pathway between the nucleus and the cytoplasm, the NPCs are critical machinery in all cells. You might remember from the title of my project, it's focused around cancer. But what does this system have to do with cancer biology? Well, an increasing body of evidence has demonstrated that many cancer cells increase their utilization of this transport system in multiple ways. For example, it has been found that many cancers have greatly increased rates of nucleocytoplasmic transfer. So this difference can be partially explained, at least by the increased number of NPC on the surface of these cancer cells. Interestingly, it has also been seen that some aggressive cancers are specifically addicted to increased transport and are unable to survive a small reduction in transport. These cancer-associated alterations suggest the nuclear transport system could be targeted in the treatment of cancer. But targeting NPCs directly was previously considered unfeasible because it was assumed that it would be too toxic to all cells. Interestingly, recent evidence has suggested that toxicity might be lessened if instead of targeting the already formed NPCs, you instead target the assembly of new ones. So as you can see here in this diagram, during interphase, as cells progress from G1 to G2 of the cell cycle, they've been shown to assemble new NPCs. The idea is that blocking this new NPC formation should leave the already formed NPCs intact, so only cells actively forming new NPCs would be affected. In support of this idea, recent evidence has shown that NPC assembly is restricted only to proliferating cells, and that post-mitotic cells that are no longer progressing through the cell cycle, they downregulate the expression of NPC proteins and do not need to assemble new ones. This idea is supported by the evidence that the core structural proteins of the NPC don't turn over in long-lived cells, such as neurons, for years or possibly even longer. Now, we all know that one of the main hallmarks of cancer cells is uncontrolled cell growth. And because these cells are rapidly proliferating, they need to be assembling new NPCs constantly. And I wanted to know if these rapidly proliferating cells are more sensitive to the inhibition of NPC assembly. To address this question, I needed to determine how to inhibit NPC assembly. I need to explain a little bit more about the assembly process to describe how I did that. So as I was saying before, it has already been established that under normal proliferative conditions, as the cell progresses through a cell cycle, it'll double its number of NPCs, as you can see here. So that when it divides, the two daughter cells will have the appropriate number of NPCs, and these daughter cells will then need to double their NPCs before they too divide. But if we inhibit this process by blocking the synthesis of essential proteins, a cell will progress through the cell cycle without doubling its NPC number, and then divide, giving its daughter cells half the NPCs it originally had. And this cycle will continue until there are too few NPCs for the cells to proliferate. But what are the essential proteins that can block new NPC assembly? To describe that, I will need to explain a little bit more about the structure of the NPC. The NPC has a well-characterized structure you can see here, which is made up of multiple subcomplexes that are in turn composed of proteins called nucleoporins or nuts. I'm not going to explain all this, but to inhibit assembly, I knocked down 28 of the 32 nuts seen here to determine which ones reduced NPCs in proliferating cells. I don't have time to discuss all those results, but I'll show how canonical members of two different subcomplexes affect NPC assembly. So in this assay, you can see I treated cells with control NUP160, NUP93 shRNA before fixing the cells for immunofluorescence. As you can see here, the green MAB414 stain for NPCs is dramatically reduced in both the NUP160 and NUP93 knockdown conditions. So this allowed me to establish that blocking expression of either of these nucleoporins could reduce NPC number, which brings me back to my original question. Are rapidly proliferating cells more sensitive to NPC assembly inhibition? And to answer that, I next looked at non-dividing and dividing cells to determine if they respond differently to the inhibition of NPC assembly. My hypothesis was that NPC depletion would only affect proliferating cells and non-proliferating cells would remain largely unaffected. So I found that in the malignant melanoma A37 cell line, under proliferative conditions, the control cells rapidly expanded while the NPC depleted cells were unable to proliferate. Conversely, if the cells were kept under confluent conditions so they could not divide, there was no difference between control and NUP knockdown cell numbers. And when I looked at these cells by immunofluorescence, I found that while the confluent cells did not have a reduction in NPCs, the dividing cells had a drastic reduction in their NPC number, demonstrating that knockdown of these NUPs reduces NPCs specifically in proliferating cells. So I next wanted to determine if these NPC depleted cells are just failing to proliferate or if they're actually dying in response to this treatment. In order to establish this, I looked at viability over time, and I found that NPC depleted cells have severely diminished viability after a few days of NPC depletion. And to confirm this result, I stained the cells after NPC depletion for the Nexin-5 cell death marker and analyzed them by flow cytometry. And Nexin-5 is on the x-axis here, and you can see that the cells had a striking increase in cell death after the NUP160 and NUP93 knockdown, and the majority of these NPC depleted cells were dead. So at this point, I had determined that these cancer cells do not proliferate and die in response to NPC depletion. I next wanted to determine if this is also true in non-cancer cells. My hypothesis was that NPC depletion would halt proliferation and possibly kill normal cells as well. To test this hypothesis, I looked at rapidly dividing normal epithelial RPE1 cell line, and I found that there was a similar reduction in the ability of cells to proliferate. You can see the knockdown cells are down here. Although if you look closely at this graph, you can see that the number of cells in the NPC depleted condition doesn't go down and instead seems to stay constant. When I directly compared these NPC depleted normal cells to the NPC depleted cancer cells from before, I found that the normal cells do indeed not decrease cell number, and that number seems to stay constant, while the cancer cells number drops significantly. So this suggested that unlike cancer cells, normal cells are not actually dying. But to directly test this, I measured viability in these and other cancer and normal cells. As you can see by these viability measures, I found that when NPC depleted in cancer cells, there's a reduction in viability, but the normal cells do not suffer from this loss of viability. This was really exciting because something that kills cancer cells and doesn't kill normal cells would be a fantastic cancer treatment. But I had found that normal cells stopped proliferating, and I wanted to determine what was happening to those cells. My hypothesis was that NPC depletion would cause a cell cycle arrest. To test this hypothesis, I measured the effect on cell cycle progression by flow cytometry. In this assay, DNA content is measured to determine cell cycle status. The distribution you see here represents a mixed population of normal cells progressing through the cell cycle. But when NPCs are depleted, there's a dramatic reduction in the cells in G2M, blue, and S phase, yellow, of the cell cycle. That data is quantified in this graph, where you can see that the NPC depleted cells are almost all in G1/G0. This demonstrates a G1 cell cycle arrest. But I next wanted to determine if this cell cycle arrest could then be reversed. So in this experiment, I depleted NPCs with doxycycline-inducible shRNA, and after the cells are no longer proliferating at day 0 here, I removed the doxy to allow NPCs to be reassembled. And I saw that the cells that were previously cell cycle arrested then begin to proliferate again. This reversible cell cycle arrest is very encouraging because if normal cells can proliferate after a temporary cell cycle arrest while cancer cells die in response to the same type of treatment, then we would be looking at an excellent cancer therapeutic target. But if this is going to be effective as a cancer treatment, it would also need to work in vivo. So I next injected A375 cancer cells into mice and measured the growth of cells as a tumor. In this experiment, I induced NPC depletion with doxy water at day 10 when the tumors were still small. And as you can see, by day 20, the NPC depleted tumors are much smaller than any of the control tumors. And when I looked at individual tumor growth, I found that all the control tumors grew much more than any of the NPC depleted tumors. And to give you a visual of how drastic this phenotype is, I had this slightly graphic image showing how macroscopically NPC depleted tumors are much smaller than control tumors. And after that, I sectioned and stained these tumors and found that NPC depletion not only reduced tumor cell proliferation, but also increased tumor cell death. And with that, I would like to finish with the conclusions for this project. First, I have identified that depleting structural NPC components reduces NPC numbers progressively only in actively proliferating cells as they divide. Second, I have found that inhibiting NPC formation with this process causes cell death in cancer and a reversible cell cycle arrest in normal cells. Third, my xenograft tumor studies have demonstrated that inhibiting NPC formation in vivo can reduce tumor burden. Finally, the major implication of this research is a basic understanding of how different cells deal with inhibited NPC formation and how that's good news for the potential targeting of this pathway for the treatment of cancer. So I'd like to finish by thanking all the people who are instrumental for this work. A huge thank goes out to all of these people and many more, and also thanks to all of you for listening. And I am excited to discuss any questions you might have.